So this is a review for exam one. Exam one is on chapter 15, the autonomic nervous system, and chapter 18 on blood. So the first thing that we talked about was the setup of the nervous system. If you took AMP1, this is probably something that you discussed in chapter 12. So we've got the central nervous system, which is the brain and spinal cord. I kind of call that like the big computer of our body. And then everything outside of our brain and spinal cord is considered the peripheral nervous system. The one thing that I like about this diagram is that it shows us the split and how with the peripheral nervous system, we have the sensory division and the motor division. And remember, I gave you that acronym SAME. So sensory is afferent, motor is efferent. The one thing that I think is missing from this diagram is the communication between the CNS and PNS. So the sensory division is going to send information to the CNS. So that's that afferent. And I always think of information is arriving at the central nervous system. And then the central nervous system is processing it and then sending the information out back to the peripheral nervous system by way of the motor efferent system. And I like to think that that information is exiting the CNS. Now, with our sensory and motor divisions, notice how we have our visceral and somatic. In AMP1, we tend to spend most of our time in the somatic. So anytime that you hear somatic, that has to do with skeletal muscle, bones, joints. And then visceral is really where we're at in AMP2. So all of those visceral gut systems like our cardiopulmonary, digestive, urinary. So we have our visceral sensory, and then we have our visceral motor division. Now, if you remember, the other name for visceral motor division is called the autonomic nervous system. And we divide up the autonomic nervous system into the sympathetic division, also called the fight or flight division, and then parasympathetic division, which is rest and digest. So most places in the body are going to be innervated by both the sympathetic and parasympathetic division. This is what we call autonomic tone, where we have this back and forth and this background rate that helps to maintain homeostasis. Now, this does not mean that every place in the body is going to be innervated equally by both sympathetic and parasympathetic. However, we are going to see most places are going to be innervated by sympathetic and parasympathetic. So the sympathetic division we call the fight or flight. And this is going to help us get ready for physical activity. So whether we're going to stand our ground or whether we're going to run out of harm's way, we activated our sympathetic division when we did the Stroop test and also when we exercised. So some of those physiological things that were happening, increased breathing, increased heart rate, your pupils were dilating to work for strategy, open up the airways, increase blood pressure. Parasympathetic division, this is our rest and digest, which is really going to do some of those maintenance things. So make sure that we're digesting our food, everything is working as it should. I also kind of call this parasympathetic division the calming voice. So after you exercised and you were resting, it was your parasympathetic division that kicked in. So that was bringing down your heart rate, breathing down your breathing, decreasing your blood pressure, also constricting those pupils. Since we're most familiar with the somatic nervous system, this is a little comparison of that innervation. So the top picture, this is the somatic motor nervous system. And here we're talking to that skeletal muscle. So that's the effector of the somatic motor division. And we have one neuron which is communicating with that skeletal muscle. And the neurotransmitter is acetylcholine. So in this case, acetylcholine is excitatory, and that's how we see that muscle contracting. By contrast with the autonomic nervous system, we actually see two neurons. And so you can see the preganglionic fibers and the postganglionic fibers. So this is one difference between the somatic and autonomic nervous system are these neuron pathways. So there's one in the somatic, and then we actually see two in the autonomic nervous system. One of the things that you want to be able to do, just like we had to on the lab quiz, is to recognize the schematic of the autonomic nervous system. So this schematic you can see is of the sympathetic nervous system, and there are some different characteristics that you can see with this picture. One, you can see that the neurons are originating in the middle of that spinal cord. This is why we call it the thoracolumbar division. And the other thing that you can notice is that the preganglionic neurons are shorter, with those ganglions being closer to the spinal cord. I think about this as thinking about if you have an emergency, you want to be a close distance from the fire department. You don't want to be living 30 miles away. The other characteristic that you can see is that the postganglionic fibers are much longer, and there's also a little more neurodivergent. So you can see that if we're in this fight or flight, it's all hands on deck. So you can see where these neurons are talking to a lot of the areas. So it's all hands on deck, making sure that we can get out of harm's way. What's not so important at this time is to be digesting what we had for breakfast or lunch. So we want blood to be going to our skeletal muscles, to our heart muscles. We want to engage those systems. Not really important to be digesting our food, so we're going to kind of suppress those areas. By contrast, we have the schematic of the parasympathetic nervous system. So here you can see the neurons originating from top and bottom. So we also call this the craniosacral division. Here you can see the preganglionic fibers are longer. The ganglion are actually farther away from the spinal cord, closer to those organs and organ systems. And we have short postganglionic fibers. So just by looking at these schematics, you should be able to note the differences. So I'm going to suggest practice drawing them out, practice writing about those differences and how you can tell the difference between them. Now, the one thing that we talked about were neurotransmitters. So what we see at all preganglionic neurons is going to be the release of acetylcholine. It's a little different when we have those postganglionic neurons. For the parasympathetic system, at the postganglionic neurons, they are going to release acetylcholine, whereas in the sympathetic, those postganglionic neurons are going to be releasing norepinephrine and epinephrine. That's mostly what we talk about with the parasympathetic and sympathetic system. Now, how these neurotransmitters respond have to do with the receptors. So actually, let me go back to this picture. So we've got the same effector tissue, the heart. So notice that we're releasing different neurotransmitters. So when acetylcholine is released and binds, that lowers the heart rate, where when norepinephrine is released and binds, it's going to increase that heart rate. So we're seeing the same effector tissue, however, because they're different neurotransmitters, they're having different responses. So the types of receptors that these neurotransmitters are binding to can also have an effect. So if we're talking about cholinergic receptors, that means that acetylcholine is going to bind to it. So there's two types that we talk about. There's muscarinic, and we would see those with cardiac and smooth muscle. Now, depending on where these are in the body, these could be excitatory or inhibitory. So it could be inhibitory, for example, on heart muscle, but it could be excitatory in like the digestive system. Nicotinic receptors, these are usually excitatory. And when we talk about acetylcholine binding to skeletal muscle receptors. It's those nicotinic receptors, and they're going to be excitatory. This is what gets that muscle to contract. So those are cholinergic receptors. Our adrenergic receptors are going to bind to norepinephrine and epinephrine. And the two that we talk about are alpha and beta. So alpha we mostly see with fight or flight. Beta 1 we tend to see with the heart, so increasing that heart rate. It also kind of suppresses the digestive system. Beta 2 we would actually see bronchodilation. So remember, if you're in a fight or flight, you need to get that air into your body, get that oxygen to your cells, so your cells can make that ATP. So we need those airways to open up, and that's what those beta 2 receptors are going to do for us. One of the things that we talked about is that we don't want neurotransmitter hanging on and binding forever to these receptors. So this is what allows these neurotransmitters to have a little bit of flexibility and a little bit of decision making. And one of the things that we talk about in AMP1 is how acetylcholine can be cleared. So it can diffuse to nearby glial cells. It can also be taken back up into the synaptic knob by way of endocytosis. There's also an enzyme, acetylcholinesterase, that will degrade acetylcholine. So that acetylcholinesterase is in that synaptic cleft. So acetylcholine can be broken down relatively quickly. Norepinephrine is a little different. So that is going to be cleared by this enzyme monoamine oxidase. And much of it is really going to be degraded by the liver. So it can actually take a little longer. So if you've ever had those experiences where maybe you had an argument with somebody or you got cut off in traffic and it tends to stay with you a long time, there's a physiological reason for that. It's because it takes a little longer for that neurotransmitter to be cleared. And that's why you're still kind of feeling those sympathetic effects for a longer period of time. One of the things that I talked about is that most places in the body, we are going to see both parasympathetic and sympathetic neurons. So we're going to see that innervation. Sometimes we can have antagonistic effects where they oppose each other, like we saw with that heart rate. One will bring the heart rate down, one will increase it. Sometimes they'll actually work together and we'll see a cooperative effect. Now, even though most places are going to have that dual innervation, it doesn't necessarily mean that they're going to innervate equally. So some places we're going to see more parasympathetic, some places we're going to see more sympathetic. So for example, in digestive organs, we would see more parasympathetic, whereas sympathetic we would see more on heart muscle. So remember, we need both of them to maintain that homeostasis, but it's not necessarily going to be equal. The last thing that we talked about is that drugs can kind of either enhance parasympathetic or sympathetic activity or inhibit it. Anytime you see mimetic at the end of these terms, that means it's going to enhance that particular activity. If you see lytic, that means it's going to suppress it. So there are certain drugs that sometimes can enhance parasympathetic or sympathetic activity. Sometimes they can actually inhibit it or suppress it. And we talked about a couple of those examples. We talked a little bit about caffeine as being one of those examples. So that is chapter 15, the nervous system. If you've taken AMP1, I know that you probably have much experience with a little bit of the setup of the nervous system. The second chapter is chapter 18, and this has to do with the circulatory system. This is really the first chapter that we're going to hit in the cardiovascular system. So first up is blood, and we talked about the functions. So number one, it's going to be transport. So this is going to be a transportation system, a highway to transport oxygen, carbon dioxide. Those are the two things that we talk about most. Nutrients, waste, and hormones. And if you remember the endocrine system, that's how hormones are going to travel. It also helps us with our immunity. And we also talked a little bit about those white blood cells that help us with immunity, also helps us with regulation. So fluid balance that we talked about can also help us regulate our temperature and pH, which is another thing that we also talked about. So we first started talking about the components of blood. So the liquid portion of blood is plasma, and that is probably the majority of our blood. So that's going to contain about 47 to 63% of our blood volume. And then we have our erythrocytes. So one of the things that we did in lab was we did a little hematocrit where we actually separate out these blood components. Erythrocytes are the heaviest, so they'll settle out first, so they will be on the bottom. Depending on what's happening in the body, males and females will also have different measurements. This is about 37 to 52% total volume. Then we have that buffy coat, which is going to be the white blood cells and platelets. And then again, on top, we're going to see the remaining volume, the majority, which is going to be that plasma. One of the things that we talked about that is important with blood is viscosity and osmolarity. Now, viscosity has to do with the thickness of blood, and we talked about how that's tied with circulation. So what contributes to that viscosity are solutes, and that's when we talked about osmolarity and tonicity. The other thing that also contributes to viscosity are proteins. So with this picture, this is when we talked about tonicity and osmolarity. So if you have a patient and you are giving them an IV, you want to make sure that that IV is isotonic with their body. So we talked about that little potato experiment, one of my favorite experiments that we do in Bio 109, where we use a potato to represent a red blood cell. And that potato, or red blood cell, is 0.9% sodium chloride. So if you are giving an IV to a patient, you want to make sure that it's that 0.9% sodium chloride, that it's isotonic with the body cells. And that's so we don't have this huge net movement of water. That's when they're going to be at that happiest state. Now, if we were to give a patient a hypertonic IV, so like 2.5% sodium chloride, there's going to be more water in the cell than out of the cell. So that water is actually going to leave and plasmolysis is going to occur. Cells are not going to like that. If we were to give them an IV of water, that would be a hypotonic solution. So there's more water outside than inside. Water is going to move in, and because our blood cells don't have a cell wall, they're going to start to swell, and then they can actually burst. They can lyse. The way that I remember that, if you look at hypotonic with that O, I try to exaggerate that O just to kind of remind myself that those red blood cells are going to swell. So that osmolarity is going to be important so that those cells are going to be the happiest, and we're not going to have plasmolysis occur. We're not going to have lysis occur. Now, this can also play a role in our blood pressure as well. And we're going to talk a lot about blood pressure in the cardiovascular system. So we did some examples of if our blood has a high osmolarity, that means it's going to have a lot of solutes. And that means that there's going to be more water in the tissues. Water is going to go from high to low. It's going to leave the tissues and go into the blood. Now the blood is going to have more volume. That's going to put more pressure on our blood vessels. That's going to increase our blood pressure. If we have very low osmolarity, little solutes, then there's going to be more water in the blood than in the tissues. Water is going to leave the blood, go into the tissues, and then we can actually have edema. So the osmolarity of our blood also plays a role in our blood pressure. And we'll talk more about that as we get more into circulation and the cardiovascular system. We also talked a little bit about those erythrocytes. So red blood cells, also called erythrocytes, are very specialized. So they lose their organelles, so they don't have mitochondria, they don't have a nucleus, so they don't live for very long, only about 120 days. And they have to be very flexible because they're going to be these very, very narrow blood vessels, capillaries that they have to fit through. So they have to be kind of springy and flexible. And they're going to be specialized to carry oxygen. And it's that hemoglobin, that big protein with the heme group, which is going to hold on to the oxygen. So most of our oxygen is going to be transported, bound to that hemoglobin in our red blood cells. Now, as a waste product of cellular respiration, we know that we are going to generate carbon dioxide. We have to get rid of that carbon dioxide. Carbon dioxide plays a role in our blood pH. So we talked a little bit about our blood pH. So it's around 7.4. Our body does not like to go below 7.35 or above 7.45. So most of our carbon dioxide is going to be transported in this bicarbonate carbonic acid system, which acts as a buffer system to help keep our pH stable. Now, remember, I like to compare carbon dioxide as an acid. As we add carbon dioxide, the blood is going to become more acidic. As we get rid of that carbon dioxide, get rid of that acid, it's going to become more alkaline. So we talked about a couple of situations where if our breathing is changed, we can either increase carbon dioxide in our blood. In that case, it's going to become more acidic. If we decrease the amount of carbon dioxide, it's going to become more alkaline. So it's this system that helps to maintain our blood pH. More to come on this in the respiratory system, because our respiratory system and our breathing is going to help to regulate this carbon dioxide level, and in turn, it's going to affect our blood pH. So this is really the first place that we're going to see that this semester. So we looked at some different scenarios of when our body would make red blood cells. So if we had inadequate oxygen, and this is what we call hypoxemia or hypoxia, you could also call it as well. When our blood oxygen gets low, it's sensed by the liver and kidneys, and they're thinking, okay, we got to make more red blood cells to carry that oxygen. So the kidneys secrete this hormone erythropoietin, which then stimulates the red bone marrow to make more red blood cells. The more red blood cells we have, the more hemoglobin, the more oxygen that's transported, and that means that our oxygen levels are going to start to rise. So this is a process that once it starts, it's going to take about 3 to 5 days to kind of ramp up those red blood cells. We also went through sort of the life cycle, sort of the life and death of erythrocytes. So we need some key ingredients to make our red blood cells. We need amino acids to make the hemoglobin. We need that iron for that heme group. We also need folic acid and vitamin B12. So those are kind of the ingredients to make our red blood cells. They're going to circulate for about 120 days. Remember, there's no nucleus, so they can't repair themselves. So then they're going to be broken apart by the liver and the spleen. We talked about how that hemoglobin is degraded. Remember, the body's going to be very efficient. So if it can use some of those parts, it's going to take them and recycle them. Any waste products we're going to get rid of. So this diagram shows us the life and death of erythrocytes. We talked about some erythrocyte disorders. So we talked about polycythemia. Poly means many, cyto means cell. So this means we have many cells, specifically red blood cells. Now, primary polycythemia, this would happen if something is going on in the body and we're overproducing our erythrocytes in the red bone marrow. Most of the time we see secondary polycythemia. This is going to increase red blood cells. So this could happen from dehydration. So we talked about that measuring the hematocrit and how it's based on that total percentage of blood volume. And if we're dehydrated, plasma levels are going to decrease. And since it is based on an overall percentage, those red blood cells are going to seem elevated. And this could indicate polycythemia. So sometimes we might see increased red blood cell counts if we're dehydrated. If the body is having difficulty getting oxygen, something like emphysema could cause this. High altitude, so we talked about if those oxygen molecules are spread out, we're not going to be able to hold on to as much. And I talked about how when I went to visit my daughter in New Mexico, that we were producing more red blood cells. And because I had more red blood cells when I returned, I actually had my best elliptical times. Because of that higher altitude, my body was sensing less oxygen, producing more red blood cells. And because I had more red blood cells with oxygen to create more ATP, I actually did my best times on the elliptical. Sometimes people will do this for training. So if they're doing physical conditioning, training for a marathon, they may go to an area of high altitude. If they're doing a lot of training, even if they're not at a high altitude, they're going to be using up a lot of that oxygen, and that could create sort of this deprived oxygen level. Now, if we have more red blood cells, this is going to increase our viscosity in our blood, and so this can cause some cardiovascular issues. So we could end up with blood clots. We could end up with slow, sluggish circulation. Sometimes it can lead to some bad things, like an embolism or a stroke. Now, the opposite is anemia. This is when we have inadequate red blood cell production or hemoglobin. There can be a lot of different reasons why somebody isn't producing enough. For example, we talked about sickle cell anemia. Could be that maybe they've got an autoimmune disorder that is affecting the absorption of vitamin B12. In any of those cases, it's that now they don't have enough and they're not getting enough oxygen. So this can affect tissues because they're not getting enough oxygen. It also affects the viscosity. And so now the viscosity of blood is going to be less. It's more like soda that's running through our blood vessels. This can cause blood pressure to drop, also our heart rate to increase, so our heart can start racing. Another thing that we talked about was blood typing, and we got the chance in lab to do some blood typing with simulated blood. So we talked about the ABO system. So if you are type A, you have A antigens on your red blood cell surface. If you're type B, you have B antigens. If you're AB, you have both. And if you are O, you have no antigens. The other thing that we talked about were the anti-antibodies, and this is in our plasma. So in our plasma, we have the opposite anti-antibodies. You do not want to make anti-antibodies towards your antigens because then you would have agglutination happening all the time. You wouldn't be able to survive. So people with type A blood, they're going to have B anti-antibodies. People with B are going to have anti-A. AB, they're not going to have any antibodies, and O is going to have both. So we talked about how this ties into blood typing. Think about if we are giving a transfusion, think about that we're only giving those red blood cells. So we want to make sure that we are giving red blood cells so that those antigens are not going to agglutinate with those anti-antibodies. The other thing that we talked about was the Rh system. So if people are Rh positive, that means that they have the Rh antigen or the D antigen. The Rh actually comes from the rhesus monkeys where it was first discovered. In lab, we were calling it the D antigen. If you're Rh positive, you have those D or Rh antigens on your red blood cell surface. If you are negative, you don't have those antigens. Now, the anti-antibodies are a little different, where you actually have to be exposed to the antigen before you start making those antibodies against it. And we talked about how this is problematic for Rh negative women that are pregnant with Rh positive babies, especially for those subsequent pregnancies, where if the mother now starts making those anti-antibodies, they can go out into the fetal blood and start attacking those red blood cells. Now, what I have also included in here is a little chart on blood type compatibility. So we had the chance in class to go through that table. Remember, anybody can give to somebody like themselves. They can also receive from somebody like themselves. However, you want to make sure that we are not giving red blood cells with those antigens that those antibodies are going to find as being foreign, and we get that agglutination. So this is very similar to that table that we actually constructed in class. So this is another source for you. We also talked about our white blood cells. We had a chance to look at these in lab underneath the microscope. So there's granulocytes that have little granules in them. These are going to be neutrophils. Neutrophils are going to be basically antimicrobial. They're going to gobble up all of those pathogens. Eosinophils help us with helminths. So they're going to blow holes in those worms, chop them up into little pieces, so those phagocytes can gobble them up. Basophils we talked about releasing that histamine and also heparin. So the way that I remember granulocytes, I think of Granny never eats bananas. Granulocytes are lymphocytes. So we talked about B and T cells. T cells are kind of like the go-between, the mediators in our immune system, very important to our immunity. B lymphocytes are going to make antibodies. Antibodies has a B in it. B lymphocyte begins obviously with B. Monocytes, they're going to help us with our long-term immunity, just like our B and T cells do. Monocytes will circulate in the blood, then they'll get into tissues and power up to these big macrophages that will gobble up pathogens. And then they can actually display those pathogen antigens on their surface and then present them to those lymphocytes. So I remember granulocytes as Auntie likes mangoes. And then if we're looking at the distribution, I gave you another mnemonic to help you remember that. Remember, neutrophils are going to be in abundance. So the little mnemonic that I use is never let monkeys eat bananas. Now, one of the things that you are going to have to do, not only in the exam, but the lab quiz, is to identify by picture the leukocytes, as well as identify erythrocytes as well. So if you like to draw pictures, you can do that. I've got many pictures in the PowerPoints, also in the lab helper, to help you with that as well. You can tell that the granulocytes, you can see those lobed nuclei. You can also see the basophils kind of look spotted, so they can kind of help you with that recognition. The other thing that we talked about was healing. So if we get a cut, we have to have a way to heal that cut, stop the bleeding. So we talked about the first stage as being the vascular phase, vasoconstriction, constrict those blood vessels so the blood isn't pouring out. Then we have the platelet phase. This is when platelets, they're going to be key in blood clotting. They're going to bind. And then we're going to have activation of the intrinsic and extrinsic pathway. These are these cascade reactions. This is a positive feedback mechanism where we have one factor goes to the next factor. Calcium is important in this pathway. Then we finally have the coagulation phase where we get that clot, and we have that fibrin where we have the little fibers, the little threads that are basically going to help to stitch up that break. And then finally, we're going to have fibrinolysis, break down the clot. We don't want this clot to keep building where now it could block off our blood vessels. And then eventually we're going to have repair. So this is a little overview of all of those pathways that we talked about. Now, I'm not going to hold you to knowing the intrinsic and extrinsic pathways. However, you should know that common pathway. So we start with that factor 10, then we go to prothrombin, thrombin, and then we go to fibrin. You should also know a little bit about fibrinolysis. So what's going to be key in breaking up that clot is plasmin. And plasmin is going to help to dissolve that fibrin polymer. I like to think of those little stitches that dissolve. That's what the plasmin is going to help us do, so that we can break down that clot. And then finally, we can have that repair and regeneration. Now, it's important that we prevent inappropriate clotting because we don't want clots to form. This could be an issue, especially if they start to travel. So one of the things that we talked about was platelet repulsion. So prostacyclin, I kind of think about prostacyclin as taking some Vaseline and putting it on a doorknob. It's going to be hard to turn that doorknob. If we kind of coated that endothelium with Vaseline, those platelets aren't going to be able to bind. So that's going to help to deter those platelets from binding. We also talked about that thrombin dilution. So with that rapid flow of blood, that's going to help to dilute it. And we also talked about some natural anticoagulants. So heparin, which is produced by basophils and mast cells, that's going to interfere with the formation of that prothrombin activator. I talked about if we were actually taking real blood in those capillary tubes when we performed our hematocrits, we would actually have to have heparin in those tubes to stop that clot from forming. So we talked about some ways that the body has to prevent inappropriate clotting. We also talked about some clotting disorders. So there's lots of different clotting disorders where people are missing maybe some of these clotting factors. Hemophilia, we talked about as being a hereditary disease. It's sex-linked, so it's recessive on the X chromosome. So it tends to be more common in males than females because males have one X chromosome. There are many clotting disorders, depending on the clotting factors. And I talked about hemophilia. I showed you that little pedigree that we saw running in Queen Victoria's line in royal families. And finally, if people are forming these clots, we may have to intervene with some medicines. So for example, we talked about Coumadin, Coumadin. These are vitamin K antagonists because vitamin K is going to be required for these clotting factors. So that's going to help that cascade not happen. Aspirin. So this is also something that sometimes people are recommended to take because that's going to suppress thromboxane to prevent those clots. And then we also talked about some other medical interventions, anticoagulants. They used leeches years and years ago to actually help to stop clots. So we talked about some of that management of clots. So that is a little overview, kind of hitting some of the high points of chapter 15 and chapter 18. As always, I always say that if you can explain it, then you know it. So if you can map things out, if you can draw them out, if you can explain it without tripping over your words or even writing it out, then you're going to be in good shape.